Unit 4 in the AP Biology curriculum. This unit is entitled Cell Communication and Cell Cycle. In this unit, we'll be learning how and why cells divide. We will also touch on the processes by which cells regulate their own division. And at the end of this unit, we will learn about cell communication, how cells respond to various signals. Today, in this introduction to mitotic cell cycle, we will cover pages 182 to 185 in the Campbell Biology in Focus textbook. Let's begin by discussing some of the reasons why cells divide. These are the roles of cell division, the reason cell division is important. The first reason why cells divide, and these are listed in no particular order, but the first one that I'm going to discuss is asexual reproduction. Asexual reproduction, as you probably know, is when there is only one parent involved in the reproductive process, and all of the offspring that are produced are genetically identical to that one parent. So asexual reproduction is easily accomplished by prokaryotes. Bacteria, for example, use a cell division process called binary fission, in which the bacterial cell copies all of its DNA and then it splits to make two genetically identical bacteria. Unicellular eukaryotes divide and reproduce in much the same way. Again, this is asexual reproduction, so a paramecium, for example, will usually make a copy of its DNA and then divide to make two genetically identical paramecia. Here in this picture, we see what was a single yeast cell in the process of splitting to make two genetically identical yeast cells. Yeast is a type of unicellular fungus. Many plants are capable of using cell division to carry out asexual reproduction. It's important to note here that plants also reproduce sexually. But I wanted to show you an example of vegetative reproduction, which is asexual reproduction amongst plants. In this photo here, you're looking at a mountainside in the Rocky Mountains of Colorado, and these beautiful trees that you see with their fall foliage are aspen trees. And genetic studies have been done in forests like this in the Rocky Mountains, where DNA is collected from a tree, and then DNA is also collected from multiple other trees within the forest. And very often, what scientists find is that the trees are genetically identical. Not just the same species, but absolutely identical. They're clones of each other. And what's happening in this case is a single tree is spreading out its roots underground. Some of those roots are sprouting brand new trees, genetically identical, and then the new trees spread their roots out and sprout new trees, and so on and so on and so on, until an entire forest of clones is created from the one original tree. Now, asexual reproduction is quite rare in the animal kingdom, but I did want to show you one example of an animal that can use cell division to accomplish asexual reproduction. Again, this is very unusual for animals, but this photo here shows a hydra, an adult hydra. Hydras are animals in the same taxonomic category as jellyfish and sea anemones. You can see that there are tentacles that are kind of growing out of the mouth area of the hydra. And what you see right here is a baby hydra, a clone, a genetically identical offspring that is growing out of the side of the parent. And here's kind of a cartoon illustration of how this works. It begins with a bud. Sounds like we're talking about plants, but this is actually an animal. And these are cells that are dividing rapidly and growing until eventually a brand new baby hydra grows from cells within the body of the parent. And eventually this hydra will separate and break off and live out an independent life, but it will always be genetically identical to its parent. So one reason why cells divide is to accomplish asexual reproduction. The second reason why cells divide is to make gametes, and that's just a fancy term for sperm cells and egg cells, for the purpose of sexual reproduction. Animals and plants use a process known as meiosis to make sperm cells and egg cells, which can then be used for sexual reproduction. As you know, sexual reproduction requires two parents and involves combining genes from those two parents to make genetically diverse offspring, so that no two offspring produced by a set of parents are going to be genetically identical, unless you have identical twins, which would be the only exception to that. So the advantage in sexual reproduction is it's dramatically increasing genetic diversity. So the first two reasons why cells divide are centering on reproduction, but of course reproduction is not the only reason why cells divide. Look at this image here. You're looking at a microscopic view. This is approximately somewhere between 100 and maybe 200 times magnification of the very tip of an onion root as it grows through the soil. And we can see hundreds and hundreds and hundreds of cells in this image. And right in this area where you see a lot of cells just densely packed together, this is called the meristem, or the meristematic tissue of the onion root tip. This is where lots and lots of mitosis cell division is taking place. So what is accomplished when these cells divide? And of course, what is accomplished is the root is growing longer, it's growing more deeply into the soil. Whenever plants grow, whenever roots grow longer, or the stems and the leaves grow taller, that is accomplished by cell division. So one of the reasons why cells divide is for growth and development. And this is true for your body as well. You started out as a single cell called a zygote, a sperm cell and an egg cell fused together to make that one original zygote. And that one cell divided to make two identical cells, which then divided to make four identical cells, and 8 and 16 and 32 and 64. And you have been dividing like that, doubling the number of cells in your body for about 15 or 16 years, or as long as you've been alive on this earth. So you have grown from a single cell to an adult, or almost an adult, with 30 trillion cells. How did you grow? How did you develop into a human being from a single cell? By lots and lots of cell division. Now, here we see an illustration of another reason why cells divide. And if you look closely at the tail of this lizard, you'll see that it's in the process of growing back. This lizard lost a portion of its tail, maybe during a fight with another animal, or maybe it escaped a predator and was almost killed and eaten, and it lost its tail. And as you probably know, lizards have the ability to regenerate or grow that tail back after it's lost. Now, you might not be able to grow a limb if you ever have a limb that's severed, but we do have in our bodies, and plants have this ability as well, the ability to regenerate or repair damage. Every time you've ever had a cut, the skin eventually grew back. When you've had broken bones, eventually the bone cells divide and replace and repair that damage, and the fracture heals. So one of the reasons why cells divide, I'm kind of skipping ahead here a little bit, is to repair or regenerate damaged tissue. And this would include healing from a cut or a broken bone. And then I skipped over this bullet here, but another reason why cells divide is just maintenance, just day-to-day tissue renewal. To illustrate that one, let me give you a very real-world example to think about. The last time you cleaned your room, and your mom sent you in your room with a dust cloth and some furniture polish, and she said, I want you to dust those bookshelves. I want you to clean your room. As you were dusting the furniture in your room, it may not have occurred to you that most of that dust that had settled on your bookshelves, guess what that dust is? That's right, it's dead skin cells. A lot of it is, not all of it. You lose skin cells by the millions each day. They die and they slough off, and obviously you've got to be making new skin cells to replace those dead, lost cells. Likewise, your red blood cells, which carry oxygen throughout your circulatory system, your average red blood cell only lives for about 4 months, and sometimes not even that long. And so you're constantly having to make new blood cells to replace the ones that were lost. That's called tissue renewal. And then if you're healing from damage, from a cut, from an injury, that would be repair and regeneration of damaged tissue. Okay, the final reason why cells divide, and maybe this is the most important or the most basic fundamental reasons why cells divide, is to increase surface area to volume ratio. What the heck am I talking about? Well, to illustrate the importance of surface area and volume and the ratio between those two, let's look at a simple example. And I know there's probably not a single living cell anywhere that has the exact shape of a cube, but I'm going to use the basic cube as an illustration, just because the cube is a basic geometric shape that we all kind of can appreciate and kind of wrap our brains around. And cells are three-dimensional, just like this cube. So a cell has length and width and height. And so it makes a pretty good model. And so let's go through and do a little bit of simple math here. Now, for the moment, let's ignore this cube, the one that's been cut into a bunch of pieces, and let's just focus on cube A and cube B. And let's assume that this is a very tiny cell, and then here is a cell that's 5 times larger. And we're going to compare these two cells. Specifically, we're going to compare the surface area of each one, and then we're going to compare the volume of each one. And we're going to talk about how the surface area to volume ratio changes when cells grow. Okay, so first of all, keep in mind, if this was a real cell, the surface area would be the total amount of area of the plasma membrane on the outside surface of this cell. So when we talk about surface area, we're really talking about plasma membrane, which is important because remember, that's how cells obtain what they need. Oxygen and water and glucose have to cross through the plasma membrane to get into the cell, either by diffusion or facilitated diffusion or maybe sometimes active transport. So the greater the amount of surface area, the easier it's going to be to obtain those nutrients. So if I was to calculate the surface area of this small cell, because it's shaped like a cube, that's easy to do. The area of one side would be length times width, so 1 times 1 is 1. But of course, that's just one side. How many sides does a cube have? Well, if you include all the sides, even the ones we can't see in this picture, the answer is 6. So if there's 6 sides, and each side has an area of 1, the total surface area of the little cell is 6. Okay. Now, volume is the amount of space inside the cell, right? So that would be the cytoplasm. That's the cytoplasm of the cell with all of its ribosomes and mitochondria and all the other parts of the cell. The greater the size of that volume, the greater the needs of the cell. If a cell has a really large volume with lots of mitochondria, then that cell is going to need lots of glucose and lots of oxygen in order to meet its metabolic needs. So if I wanted to calculate the volume of this small cell, it's length times width times height, so 1 times 1 times 1. That's easy math. It's 1. So the surface area is 6, the volume is 1, and if I look at the ratio between these two, that would be a 6-to-1 ratio. You could also do that by dividing. 6 divided by 1 gives me a ratio of 6, or a 6-to-1 ratio. Now, that doesn't mean a whole lot unless we compare it to the cell that's bigger. Right beside this little cell is a cell that is 5 times larger. Okay, notice it has lengths of 5 on each side instead of just 1, so it's 5 times larger. Now my question to you is, obviously this cell is going to have a bigger surface area, and it's also going to have a bigger volume. But my question is, when a small cell grows into a larger cell, which number grows at the fastest rate? Does surface area grow faster? Does volume grow faster? Or do they grow at the same rate? You might assume that they would grow at the same rate, but let's do the math and find out. So the total surface area for this cell would be 5 times 5, that's 25 for one side, but remember we have 6 sides, so 25 times 6 is 150. No surprise, the surface area of this big cell is a lot bigger. It has a lot more plasma membrane on the outer surface than the little cell. That's not a surprise. The volume is also going to be bigger. Length times width times height, 5 times 5 is 25 times 5 is 125. Now my question a moment ago, remember, was which of these numbers grew the most? Did the surface area increase the most? It went from 6 to 150. Or did the volume increase the most? It went from 1 to 125. So let's think about this mathematically. How many times bigger is this number compared to this number? Well, 150 is 25 times larger than 6. So the surface area, when the little cell got bigger, it got 5 times bigger, the surface area got 25 times bigger. How much did the volume grow? How much larger is this number, 125, compared to this number? It's a big difference, right? 125 is 125 times greater than the original volume of 1. So what does that mean? It means that when a cell grows larger, the volume grows much, much more dramatically larger than the surface area. Volume increases at a much faster rate than surface area. Well, that's real important for cells because what it means is that the needs of the cell, the total amount of water and glucose and oxygen and other nutrients that the cell needs, those needs are going to grow 125 times larger, but the ability to get what the cell needs is only growing 25 times larger. In other words, the needs of the cell has grown faster than the ability to get what the cell needs. So this turns out to be one of the most basic fundamental reasons why cells are small. There's a reason why we need a microscope to see most cells. It's because only the tiniest of cells are going to have a healthy surface area to volume ratio. And the larger the cell gets, the smaller the surface area to volume ratio. It seems kind of like a contradiction, or it's a little bit counterintuitive, but you need to remember that the larger the cell, the smaller the surface area to volume ratio. And so if you're a big cell and you're having a hard time surviving because you can't get enough nutrients quickly enough, if you're a large cell and your surface area to volume ratio is too small to keep you alive, what's the only way to fix that? How can a large cell fix that problem? If you said by dividing, you are right. This large cell can divide to make 2 smaller cells, and if it's still too big, it'll just divide again and again and again. So here's what this same cell would look like if it has divided a whole bunch of times. So instead of one big cell, now you have a whole bunch of little tiny cells, and you see the result? We're back to a very healthy surface area to volume ratio. Now don't be confused by the math. The bottom line is quite simple, and this is your extra credit phrase that pays for this week. Small cells are healthy cells. Small cells are healthy cells. Here's another way to look at the same thing. Smaller cells have a greater surface area to volume ratio. That means a tiny cell is going to have a much easier time obtaining glucose and oxygen and water, whereas a large cell is going to have a very difficult time getting enough of the things that it needs to survive. So altogether, that was 6 different reasons why cells divide. Asexual reproduction, making gametes for sexual reproduction, growth and development, tissue renewal or maintenance, tissue repair, that is repairing damaged tissue, And finally, cells divide to increase their surface area to volume ratio. So let's look at some examples of cells in the process of dividing. In this picture here, we see a single amoeba, a unicellular eukaryote. It has made a copy of its DNA, so there's a nucleus here and a nucleus here that are identical. They have the exact same DNA. Now the amoeba is splitting to make two identical cells. Here we see a bone marrow cell that is dividing to make two identical cells. This is how new blood cells are made. And in the picture at the bottom, we see how your life began. This is a single cell called a zygote that is in the process of dividing to make two genetically identical embryonic stem cells. And right now it's going to be two cells, but then those are going to divide to make 4, and then 8, and then 16. And for a while you were just a little ball of identical cells until those embryonic cells began to differentiate and specialize to make skin tissue and blood tissue and muscle tissue and organ tissue. But this is how your life began, and to a large extent, this is what you've been doing for the last 15, 16 years, is lots and lots of cell division as you have grown and developed into the adult that you are today. So let's go through some basic terminology that's going to come up a lot in this unit. First of all, genome. A cell's genome is the complete endowment of DNA within the cell, and the genome contains all of the genes needed to code for the entire organism. Now that's an important point. That means you could take a skin cell, an epithelial cell from your arm, and in that arm cell are the genes for teeth and eyeballs and liver and kidneys. Even though it's in your arm and it's just a skin cell, every cell in your body has the exact same genome. And so we've learned a lot about the human genome in the last 15 to 20 years. We used to think there were about 100,000 genes necessary to code for a human being, but now we know that it's a much smaller number. It's a surprisingly low number, about 20,000 genes in the entire human genome. These are carried on 23 different kinds of chromosomes, and you have 2 of each one of those chromosomes because you inherited 23 chromosomes from your dad in the sperm cell and 23 chromosomes from your mom in the egg. And so you have these 20,000 genes that are carried on 23 pairs of chromosomes. Somatic cells are regular body cells, skin cells, liver cells, kidney cells, heart cells. Every cell in your body is a somatic cell except sperm and egg. Gametes, as we mentioned before, is just the fancy term for sperm and egg. Gametes are reproductive sex cells that are haploid. And you'll learn more about the meaning of this word later, but essentially haploid is an adjective that means that the cell has only one set of chromosomes. A haploid cell has half as many chromosomes as a regular body cell would have. Mitosis, we're going to be talking a lot about mitosis in the near future. Mitosis is the division of the cell nucleus. The purpose of it is to separate the duplicated copies of all of your chromosomes. And the purpose of mitosis really is to produce 2 nuclei or 2 copies of the nucleus that are genetically identical. So mitosis is the division of the nucleus. Cytokinesis is the splitting of the entire cell, all of the cytoplasm separating to physically make two separate cells. One of the confusing parts about this unit is there are a lot of words that sound alike. Chromatin, chromosomes, chromatids. It's enough to drive a biology student crazy. But basically, just to get us started down that road, chromatin, this term here, is the stuff that chromosomes are made of. Chromatin refers to long, thin, relatively uncoiled or very loosely coiled fibers of DNA. And these DNA fibers are wrapped around tiny little spools inside the cell called histone proteins. And this is a hard concept, I think, to try to illustrate, but the textbook illustrator was attempting to do just that. So here they're showing us the DNA double helix, which we're all familiar with, and it's an incredibly long molecule. Just to give you an idea, every cell in your body has approximately 2 meters of DNA. That's 6 feet of DNA. Now think about that. A single microscopic skin cell that's so tiny you have to have a microscope to see it has about 6 feet of DNA inside of it. How's that possible? Because the DNA doesn't stay straight like this. The DNA is wrapped again and again and again around these tiny little round proteins called histones. They're like little spools, and the DNA is like the thread that's wrapped around the spools. And as that DNA wraps around those histones, it can twist and coil and wrap around itself. And as the DNA wraps and coils and shortens, eventually the DNA can condense into a solid mass that's visible with a microscope called a chromosome. So maybe it's useful to kind of think about this in reverse. If we take this picture of a chromosome, here's an actual photograph right here of a chromosome from a single human cell. This is just one of 46 chromosomes in the cell. If you take that one chromosome and you zoom in on this little area right here, you'll see just feet of DNA wrapped and coiled and folded. And if we zoom in on that, we can see that the DNA is folded back and forth and back and forth, and all of that DNA is wrapped tightly around these little histone spools. Well, sometimes the DNA is in a relatively long, thin, uncoiled state. We would call that chromatin, but when it coils up very tightly to form a chromosome, then that's what we call it. We call it a chromosome. And I know that's confusing, but technically, the DNA only takes on the chromosome configuration during mitosis, when the cell is actively in the process of dividing. But we'll spend more time talking about that later. What is a chromosome? In short, it's a gene-carrying structure found in the nucleus of our cells. And what is it made out of? It's made out of chromatin. The chromosome is just a highly coiled strand of chromatin. It's DNA that has twisted and coiled and shortened and thickened. And how many chromosomes do we have in our human cells? We have 23 pairs of them in all of our somatic cells. And of course, that means 46 chromosomes total per somatic cell. Somatic cells, remember, are just normal body cells. Okay, let me throw one more definition at you. Chromosomes in our somatic cells, our body cells, chromosomes occur in pairs. And here the chromosomes are color-coded to show that when you have a pair of chromosomes, one of those chromosomes was inherited from your father, and the other chromosome was always inherited from your mom. Of course, our chromosomes are not really color-coded blue and red. That's just a convenient way to show it in the illustration. But for every chromosome that was passed down to you in the sperm cell, there is a corresponding chromosome that was passed to you in the egg. And we refer to these pairs of chromosomes as homologous pairs. This is a homologous pair of chromosomes. What does that mean? It's a pair of chromosomes with the same size and the same shape, and they both carry genes for the same types of traits. So if this chromosome has a gene for being tall, the other chromosome will also carry a height gene. It might not be the gene for tall, it might be the gene for short. If this chromosome carries the gene for brown eyes, this chromosome will also carry an eye color gene. It won't necessarily be the same gene. It might be the gene for blue eyes or green eyes. So these two chromosomes are not genetically identical. I repeat, these homologous chromosomes are not genetically identical. They cannot be, because remember, you inherited one of them from your father and the other you inherited from your mother. And hopefully your parents were not close relatives. I think that's safe to assume. So of course they're not going to have exactly the same DNA, but they are the same size and they are the same shape, and they do carry genes for the same kinds of traits: height, eye color, hair color, skin color, whatever the genes happen to be. Okay, we're almost finished with this lesson. The last thing I'd like to talk about is what is known as mitotic cell cycle. This diagram essentially shows you the events, the major events in the life of a cell. So let's begin by just looking at one cell. Just ignore this other cell for a moment. Let's just look at the one cell. This could be any somatic cell in your body, a skin cell, a lung cell, whatever you want it to be. And the first thing that this cell is going to do is it's going to enter into a period of time known as G1, in which intense growth is going to take place. I always think of the G in G1 as standing for growing. That's not really what it stands for, but that's a convenient way to remember it. So this tiny cell will grow physically larger for a period of time, roughly the first third of its life. And then once it reaches this dividing line and enters into the S phase, that growth slows down quite a bit. So most of the growing takes place during G1. The second phase in the life of a cell is called the S phase. And you can see that S stands for synthesis. And the main thing that happens during S is that all of the DNA, all of the chromatin, or the DNA fibers within the cell, are copied. They are replicated. An exact copy is made of all of the DNA. So the cell is not growing as rapidly during the S phase. Remember, it did most of its growing during G1. Instead, the focus during S is making a copy of all of the DNA. So by the time the cell reaches the end of S, that cell actually has twice as much DNA as when it began the S phase. The DNA is literally doubled as it is copied during S. Next comes the G2 phase. Oops, I forgot that I put this. I wanted to show you with this illustration. I confused myself for a moment, but I wanted to show you what your chromosomes look like before S. So if you could see the chromosomes in your cell during G1, they would look like this. This is just looking at one homologous pair of chromosomes. Each chromosome would only have one copy of itself. But after S, when we get to the end of S, now look at those chromosomes. Each chromosome now has two copies of itself. So that's that copying that I was talking about. You can see the amount of DNA literally doubles. And the two copies, the two identical copies of the DNA for each chromosome, those are called sister chromatids. So this is what your chromosomes look like before S, and this is what your chromosomes look like after S. So just to review, the cell grows and makes a copy of all of its DNA. And then G2 is when the cell is getting ready to divide. It's making preparations for the division process. So I always think of the G2 as representing getting ready. That's not really what it stands for, but that's just a convenient way to remember it. Now, before we talk about mitosis and cytokinesis, I want you to notice that all of those periods of time that we just went through, G1, S, coming all the way around G2, that whole period of time collectively is referred to as interphase. Interphase is by far the longest period in the life of a cell. Most of a cell's life is spent in interphase. Interphase means a couple of things. It means that the cell is not currently dividing. And secondly, interphase is when the cell is carrying out normal cell activities, whatever that is. You know, if it's a pancreas cell, it's busy making insulin. If it's a nerve cell, it's busy sending messages. If it's a stomach cell, it's busy making acids and digestive enzymes. It's doing its normal job. But in the meantime, while it's doing all of those normal activities, it's also growing, copying its DNA, and getting ready to divide. At the end of interphase, or right after interphase, comes a relatively brief period of time known as mitosis. We're going to focus on mitosis in the next lesson. Mitosis is the division of the nucleus. It's splitting all of the chromosome copies apart so that the cell ends up with two identical nuclei. And that is followed by cytokinesis, which is the physical splitting of the entire cell so that after mitosis and cytokinesis are complete, we no longer have just the one cell. We now have two identical cells. The nucleus inside each of these nuclei, the DNA inside each of these nuclei is absolutely identical. Okay, so just to recap, interphase is when the cell is not dividing. Interphase is made up of three subphases. G1 is when the cell grows. S is when all the chromosomes are duplicated or copied. G2 is when preparations for mitosis are being made. Remember, G2 is when the cell gets ready to divide. Mitosis is the division or splitting of the nucleus, and you're going to learn next time that mitosis is made up of 4 distinct phases: prophase, metaphase, anaphase, telophase. Cytokinesis is the division or splitting of the entire cell so that one cell physically splits into 2 identical cells. Before we leave the topic of mitotic cell division, I want to mention G0. Some cells, after dividing many times, after this cell cycle goes around and around and around a bunch of times, sometimes the cell will enter into G0. And that means the cell is still alive, it's still fully functional, it's just not dividing anymore. It's no longer copying its DNA, it's no longer getting ready to divide, and it's no longer carrying out the process of mitosis and cytokinesis. It's like the cell kind of just pauses and no longer continues to participate in cell cycle. So this is referred to as G0. As I said, it's an extended period in which the cell basically exits from cell cycle and no longer divides. For some cells, this is temporary, and the cell may, in the future, it may return to normal cell cycle, normal division, depending on the needs of that particular type of cell. But in the case of brain and spinal cord cells, at least most brain and spinal cord cells, once you reach adulthood and your brain and spinal cord have reached their full adult size, most of those cells go into a permanent G0, which means you're no longer going to be able to make any additional brain or spinal cord cells. They're no longer going to divide. This is why it's so difficult to fully recover and heal from a brain or spinal cord injury. If we have a cut on our skin, those cells will grow back pretty quickly to heal that wound. But that's not the case for traumatic brain and spinal cord injuries because those cells are in G0. Well, guys, this brings me to the end of our first Unit 4 lesson. I hope you've learned something, and I look forward to seeing you soon. Take care.